behalf of Ionis, of what we've done recently. But like everybody else before me said, this really, takes a team. I'm just here in front of you, but this is the work of many people behind the scenes. So before I go on, I think we've all heard this in many different analogies today, but you know that myotonic dystrophy is caused because you have 3 nucleotides that are repeated a number of times, too many times, and those repeats will stick to muscle-blind, or muscle-blind will stick to those repeats, and as Matt said, then muscle-blind cannot go and do the things it's supposed to be doing. And then this then leads to a whole slew of various symptoms that you're all having. So anywhere symptoms from gastrointestinal, obviously myotonia, muscle weakness, but then also CNS issues. So why did Ionis become interested into myotonic dystrophy a while back? And it did because it's been called a toxic RNA disease. And what Ionis does well is target the RNA. And so we thought that this was really a disease that we could try and target and make a difference in. It was also a disease, a rare disease, and a disease that had a high unmet need. And so you all needed somebody to try and find a cure for it. So trying to explain to you, especially the newcomers, others have seen me give some of this presentation before, so they might understand the mechanism of action of our antisense oligonucleotides. But for newcomers or people who have never heard me talk, just wanted to give you a refresher on biology 101. So your gene gets transcribed into RNA, and then your RNA gets translated into a protein, and then, proteins do what they do. Sometimes they do bad things, sometimes good things. In your case, it's really the RNA that's the problem. But antisense oligonucleotides are little pieces of synthetic DNA that we can build in the lab and target specific areas on the messenger RNA. And so because it's a code of 4 letters, we know the code, and we can make specific pieces of what we call ASOs, or antisense oligonucleotides, that will then go link to that messenger RNA, and then that causes destruction of the messenger RNA, and then that protein doesn't get produced. So here you will see a diagram of the molecule that we had developed and that actually went into the clinic, and I'll go over that. So it has a piece of naked DNA in the middle, so that is the DNA that where we know the code, and then we put specific chemical modifications on each side of that DNA. And those chemical modifications will increase the affinity of the drug, increase the tolerability of the drug, and increase the potency of the drug. And so this is what we call generation 2.5. And so what it does is that little blue piece, I'm not sure where the pointer is, I don't know if you have one, anyways. So you see the little blue piece is our antisense oligonucleotide, will bind to the RNA, and those little red dots are muscle-blind molecules that are stuck there. And then that duplex will bring in an enzyme that will then destroy the RNA and release the muscle-blind, and then muscle-blind can go do what it wants. So we tested this drug, obviously, in animal models in mice, and various different mouse models, and then also tested it in monkeys, and then we took it into the human. And so we had a phase 1 trial, which is a safety trial, and phase 1 trials are often divided into single ascending dose studies and multiple ascending dose studies. So basically, a single ascending dose study is a study where a patient receives 1 injection of the drug, and in this case, those drugs were given subcutaneously, so just under your skin. And then you follow these patients for 28 days, and if everything's fine, then you can go on to the next dose. So you have to treat 4 patients in 1 dose level. You look at that data, everything's good, and you go to the next dose, et cetera. And that's how you can also try to better understand how high you have to go, because you look at the amount of drug that is afterwards in the blood after 28 days, and you can, based on that, kind of determine which amount of drug you need to treat patients. So that study was completed in 2014 and showed good tolerability, and that was done in the UK. And then from there, we moved into our patient study. And so the patient study was what we call a phase 1/2A. It's still a safety study mainly, and it was done in myotonic dystrophy patients, and I know that a few of you guys in the audience here participated in the trial. So there we followed 48 patients, and again, here it's called a multiple ascending dose study. So each patient got multiple doses of the drug, but we had to go through the first dose level first. So in the first group, 8 patients got 100 milligrams. Once we saw that 100 milligrams were safe, we can go to 200 milligrams, 300, 400, and then 600. Those were the doses that we tested. And just to show you on the schematic on the bottom what these patients had to go through in the trial. So they had to be screened, and then they would get a muscle biopsy. That's your little red star. So there was a muscle biopsy in the tibia. And then on the first week, they got 3 injections. So normally it was a Monday, Wednesday, Friday. And then they would get an injection every week thereafter for 6 weeks. 2 weeks after the last dose, they would get another muscle biopsy, the same tibia, so the same side, but a little bit spaced out or like an inch away from the first biopsy. And then they would still come back to the clinic for 3 visits over 14 weeks so that we could do various different assessments with these patients. So they did a lot of muscle testing. So the primary objective of the study was, as always with phase 1, phase 1/2 studies, is safety and tolerability. And so that would be done by looking at labs and looking at ECGs of patients. And it was really trying to see how tolerated the drug is. Secondary objectives, we look at pharmacokinetics, oftentimes in the blood and in the urine, and that's basically how the drug is distributed throughout the body, and do we get to the right levels of drug levels in the body. And we also tried to do it in the muscle tissues. To start out with the study, we weren't sure we could actually look at drug levels in the muscle tissues. We weren't sure we were going to have enough muscle tissue to look at splicing changes and drug levels, but it ended up that we did. So we looked at all of that. And then some of the exploratory objectives were the clinical outcomes. And so patients did 3 different tests of myotonia. They did a whole bunch of muscle strength tests, and then they also did functional tests like 6-minute walk and so on. So I talked about some of the functional outcome measures last time, and so I won't go over that, but one of the points that I also made last time is that when we looked at the pharmacokinetics, so the level of the drug in the muscle, we realized that we really were not hitting what we needed. So to orient you on the slides, on the bottom horizontal axis, you have the different doses that we tested in the human, so 100, 200, 300, 400, and 600 milligrams. And then on the vertical axis, you have the concentration of the drug in the muscle in micrograms per grams. Basically, we needed to hit 10 micrograms per grams in most of the patients because that would get us, 50% of what we wanted to see. And as you can see, the graph doesn't even go up to 10 micrograms per grams, right? So we had a few patients in the higher dose cohort that had a good amount of drug in their muscle, but the majority of the patients were just too low. And so that's what really took us to go back to the drawing board and try to develop a more potent drug, because we knew that even if you treat it for the longest time, you just were never going to get to the level of efficacy that you were going to need. Now, having said that, we've done a lot of work on still the rest of the muscle tissues that we had. And so this is new data that I'm presenting. And so we were looking at all the splicing biomarkers in some of those pieces of muscle tissue. And I'm presenting data here of 29 patients. So we didn't have enough muscle biopsies, from the day 1 to the day 50 in all 48 patients, but we had it in the 29 patients. And what you're looking at here is a composite index of 22 splice events. And so again, when you go from left to right, you have the different groups, the placebo, 100, 200, 300, 400, and 600 milligrams. And what you see on the bottom is before treatment, and on the y-axis after treatment. So the line that diagonally goes from 0 to 1, if people were clustered on the bottom towards 0, that means that they were normal. So there's really nobody that's clustered around the 0. And the higher you go towards the top of that line, the more severely affected patients are. So you can see that patients were, in terms of their splicing profile, right? So you can see that they were heterogeneously affected, as we expect in myotonic dystrophy. And there was really no change from before to after treatment, except in 2 patients that you see in the 600 milligram dose cohort. And those were the 2 patients towards the lower part of the graph, right? Because whatever goes low is better. So basically, there seemed to have been an effect of the drug on the splicing biomarkers from those 2 patients from before treatment to after treatment. So now you may say, well, this is an overall splicing event. This just combines everything. Well, we also looked at the different splice events for each of these patients, for these two patients. So these are the graphs at the bottom, and the top are two different patients. And as you can see, horizontally, it lists all the different splice events that we looked at. And those different splice events make up the composite index. And so to orient you on the slide, the grayish bars that are on the top and that go towards the end, that's really the range of the splicing profile in a set of normal people, so who do not have myotonic dystrophy. The purple, or the pinkish, is the range of what happens in people with myotonic dystrophy. So that's their aberrant splicing, basically, in those different analytes. And then what the arrows show, so the bottom of the arrow shows where the patient, those two patients that I showed you, where they started, so what the splicing was in their first muscle biopsy, and then pointing upwards was where they ended after the treatment. So basically what this shows is that the change in their splicing profile went towards normal, or towards what is seen in normal patients after a treatment. So this really, I think, shows that the splicing biomarkers are really something to look into. Now, you might also say, well, you know, this could just be two different biopsies 8 weeks apart. Maybe it's just variation. So the Rochester group actually has really nice longitudinal data where they actually compared the first biopsy to the second biopsies in some patients. So they had 116 patients where they did two biopsies in the same leg in the patients, either at 8 weeks or at 12 weeks apart. And so this is the general variation that you see in those patients. So again, on the bottom is first biopsy versus second biopsy. So that's basically how they lie. And if you superimpose our two patients in purple, and really you should compare the purple dots to the red dots because that's the same time frame, you actually see that they are coming outside of what you could think is normal variation in splicing that you could see between two biopsies. So the point I really want to make here is that I think, you know, for the science community, it's really important to look at splicing changes, and I think having a battery of splice event will be important for future trials. So really, in terms of conclusions of this trial, so the drug was well tolerated. Nobody discontinued the study. I know for you guys, the study wasn't successful. For people who run trials, it was successful in many ways because we were able to show that we could run, you know, this trial at 8 different centers across the US, that, you know, 8 different centers were able to do muscle biopsies in the same manner, and that we could analyze the tissue and we got good results. We could also do all the physical therapy assessments at 8 different centers and, you know, get good data. Now, the only problem was that 600 milligram dose did not give us a robust effect in the majority of patients, and we obviously were too low in dose. And so, you know, 600 milligrams, you could tell me, well, you could have gone to 800 or 1200. Well, 600 milligrams was already 3 injections of 1 mil, or 1 injection of 3 mils, which is quite painful, so you wouldn't want to do that. And it would be, you know, at a weekly time point. So it was really better at this time point to go to get a more potent drug. But it is still, you know, we are still mining the data. We're still looking at anything we can in all the tissues and samples that we got, because we feel that, you know, it's our responsibility to make sure we get all the information out of this and that we can share it with the community. So what's our focus now? So our focus has been on trying to find a better molecule for the muscle. And what we've really been trying to do is find a ligand that can bring our antisense oligonucleotides to the muscle specifically. The first molecule we had, we just injected it under the skin and kind of expected it to go to the muscle, and it just doesn't go to the muscle very easily. And if it did go to the muscle easily, we would probably not have been the first company to have a trial in myotonic dystrophy. So, but we know we can find ligands to really try to target our ASO to specific tissues. We've done that, and we're working on it for different organs. So we know we can target the liver using this ligand called GalNAc, and GalNAc can use the ASAG receptor to go specifically into the liver. And so basically what we're showing here in the graph is that the blue line, that would be the ASO without the ligand, and that's its dose response curve. Once you attach the ligand to it, you increase the potency 30-fold as the line moves to the left, and so you get more drug into the liver. The same thing can be done in terms of trying to specifically target beta islets of the pancreas, and that is by using a GLP-1 ligand. And so, the same similar graph to before, if you just look at the dose-response curve, you can shift it to the left, making the drug more potent. And the images just show you the immunohistochemistry images on the top, just show you the brown stain would be our ASO. And so once you put the GLP on mallet ASO, you get more of the drug within that blue line, which would be the beta islet of the pancreas. And then you also get more knockdown, which is the graph below. So we know we can do it, it's just trickier to try and find the ligand for the muscle. What we're also focusing on is on the central nervous system. And so I think over the last couple of days here, everybody really has been talking about the importance of central nervous system, and I mean, this has been going on for quite a few years actually. So we're working on the muscle, but we're also focusing on the CNS. And one of the reasons is also because we've had recent successes at Ionis to target the CNS. So we've developed a drug for spinal muscular atrophy that's on the market now. We've had good preliminary results in our phase 1 trials with Huntington, and our ALS trial seems to be going on fine. Now, the only thing the community needs to realize is that if we go for, or if Ionis, with our platform, goes for a drug into the CNS, it will have to be delivered via a lumbar puncture, which we don't think is a problem. It normally then could be done, either on a monthly basis or on a 3-monthly basis, and we have treated, from infants to adolescents to adults to elderly. So we know we can do the procedure in, every patient population. It's just a matter of whether the patients would approve a lumbar puncture. So the path to develop a drug into the CNS, obviously a lot of people are working on CNS mouse models, and we will be needing to collaborate and work on those to look at target engagement in the mouse. We would need biomarkers in the cerebrospinal fluid to be able to confirm target engagement. So any researchers out there who would like to work on CSF biomarkers for myotonic dystrophy, it's the time. We're happy to collaborate. And then we also need to work on clinical endpoint. We know the FDA really wants to hear from the patients, want to know what the patients suffer the most from and what would really be meaningful for them. So there's a lot of patient input that we have all already received or that's already out there. MDF was the first MDF in 2016 set up the first external, there we go. The first externally led patient-focused drug development meeting that that resulted in a Voice of the Patient report that is really helpful. Also last year, the CNS roundtable that was done at this meeting was extremely helpful, I think, for a lot of people. And I know that for me, there was a lot of things that I did not realize what affected patients, and I think it was really important for us to hear it. And so we're looking forward to that publication. And then the Christopher Project report also provided a lot of information for my industry partners and myself, and I think also for researchers to try to focus on what's important for the patient. So what we are trying to do is we really want to focus by putting all together all that information that's out there on what the patients are reporting, what their symptoms are, but also trying to figure out what's the most important symptom, what impairments, what improvements, would you expect from a drug that would affect the CNS aspect? I know we always talk a lot about what the problems were, but what would you expect to see changed? And what types or what magnitude of improvement are you expecting? And what kind of benefit-risk ratio would you accept as a patient population or as individuals? So over the summer, we had an intern at Ionis come, and we worked on a survey to try and better understand that, so better understand the clinically important aspect of all those symptoms for the patients. And we actually had tried the survey on a few local patients, and then took the feedback we got from the survey and the answers and tried to refine this. And so we are expecting to send out the survey shortly through the MDF website, and so be on the lookout for it, and please participate. This will be really valuable information for us to have. And then we're also going to try and do a parallel caregiver survey. So in conclusion, you know, that's where we are. So we are still mining through the data that we obtained from our clinical trial. I know it's been a while since the clinical trial has completed, but we are working on a publication because we feel it's important to get out there, even though if, you know, technically some people would say the trial wasn't a positive trial. We're still focusing on the systemic therapy for patients, but we are also now investigating the CNS aspects of the disease. And with that, I just wanted to thank all the sites who participated. Everybody at the sites, you know, really worked very diligently. But far and foremost, thank you to the patients who participated in the trials, because we couldn't have done it without you. So thank you.